Hi everyone. So in this video we will study about one of the very important topics in virology, that is human immunodeficiency virus, or it is also called as HIV. So HIV virus, it is a causative agent of a disease called as AIDS or acquired immunodeficiency syndrome, which is considered as the biggest threats of last 3 decades because this HIV virus can cause immunodeficiency in an individual who is infected, which means it suppresses the immunity of an individual, which results in development of various opportunistic infections, as well as it is almost impossible to completely cure this AIDS once the person turns completely develops the final stage of the disease, that is AIDS. And also there are no vaccines available currently. This HIV virus belongs to the family of Retroviridae. So there are special group of viruses called retroviruses. So this HIV is actually a RNA virus. These retroviruses has a special capacity of converting single-stranded RNA into double-stranded complementary DNA because of the enzyme reverse transcriptase. So this single-stranded RNA, after entering into the host cell, it converts into double-stranded DNA because of the enzyme reverse transcriptase, so that it can easily incorporate with the human DNA and cause infection. So this HIV belongs to the genus of lentivirus, which involves HIV-1 and HIV-2. So next, coming to the morphology of HIV. So it is roughly spherical or round in shape, which measures about 80 to 120 or 110 nanometer in size. It is a medium sized virus. So it is round in shape and it has an envelope. So HIV is an enveloped virus. So you can see here outermost covering, that is envelope. So this envelope is lipoprotein in nature, which means it is made up of lipid plus protein. So lipid part of this envelope of HIV virus, it obtains it from the host cell membrane. So once the virus enters into the host cell, maybe of animal or human being, it acquires this lipid part from the host cell membrane. And the protein part of the envelope, it has two components, that is glycoprotein 120 and glycoprotein 41. You can see here glycoprotein 120, which is a knob-like or mushroom-like structure, and here you can see the glycoprotein 41, which is rod-like structure. So the main function of glycoprotein 120 is attachment. So once the virus enters into the body, it has to attach to the host cell, right? So it binds with the CD4 plus T cell receptor on the host cell so that the virus enters into the cell. And the glycoprotein 41 is a fusion protein. After attachment, it helps in the fusion of the virus with that of the host cell. So these two proteins plays important role in pathogenesis of the virus. So next, coming to the nucleocapsid. So next to the envelope, that is outermost part of the virus, there will be nucleocapsid. So next to envelope there will be capsid, which is icosahedral in symmetry. Inside that there will be a cylindrical core, which encloses two identical copies of positive sense linear single-stranded RNA. And also there will be 3 enzymes, viral enzymes. First one is reverse transcriptase, integrase, and protease. So this is about the structure of HIV virus, which is spherical in shape and measuring about 80 to 110 nanometer in diameter, and it has envelope. It is an enveloped virus. Envelope is made up of lipoprotein, that is lipid part, and protein part, which has two components, that is glycoprotein 120 and glycoprotein 41. and inside the envelope there will be capsid, which is icosahedral in symmetry, and innermost layer will be, and there will be a cylindrical core which encloses 2 identical copies of single-stranded RNA, and also there are 3 enzymes, that is reverse transcriptase, integrase and protease. Thank you for watching.